Good morning, Assalamu alaikum, dear researchers, presenters, and participants. On the behalf of the Applied Zoological Society of Pakistan, with the collaboration of Quaid-e-Azam University, Islamabad, I, Mudassir Hassan, PhD scholar from Gormi College University, Faisalabad, warmly welcome all of you in the 4th International Conference on Applied Zoology 2021. This is our first session of today's conference. Here, it comprises the talks from the field of biochemistry, genetics, molecular biology, physiology, microbiology. I will moderate this session. So let's start our session with first presentation by Dr. Shgufta Saeed from University of Veterinary and Animal Sciences, Lahore. She will present her topic on production of gallic acid under solid-state fermentation by utilizing waste from food processing industries. Please, Dr. Shgufta Saeed.
Bismillahirrahmanirrahim. I welcome you all in the International Conference on Applied Zoology. The topic that I am going to discuss today is related to the production of gallic acid under solid state fermentation by utilizing waste from food processing industries. Myself, Dr. Shagufta Saeed, and I am working as an assistant professor in Institute of Biochemistry and Biotechnology, University of Veterinary and Animal Sciences, Lahore. Starting from the introduction of gallic acid, it is a phytochemical compound present in various parts of plants as a secondary metabolite. It's colorless, odorless, slightly yellow or crystalline compound. Its molecular formula is shown here. Melting point is approximately 252 degrees centigrade. It has a wide range of applications in the pharmaceutical industry as antioxidant agent, antibacterial, anti-cancer, precursor of antibacterial drug trimethoprim. In food industry, it is used as food preservative and also used in the preparation of high-grade leather tanning in the manufacture of inks, paints, and color developer. Gallic acid is produced by hydrolyzing tannin either by the chemical hydrolysis or the enzymatic hydrolysis. Most of the microorganisms they have the enzyme called tannase that can hydrolyze tannin to gallic acid and glucose. Tannins are naturally occurring water-soluble polyphenols, secondary metabolites found in various parts of plants such as wood, bark, leaves, fruit peels, seeds, and the galls. This table shows the examples of various microorganisms that have the potential to produce tannase, like from bacteria, lactobacillus, streptococcus, enterococcus, then yeast like Pseudomonas, Candida piscea, from fungi, it could be Aspergillus, Trichoderma, and so on. Statement of the problem is that conventional production of gallic acid by chemical hydrolysis of tannic acid is unfavorable due to poor yield, high cost, and discharge of little effluent that cause environmental pollution. Hypothesis of the present study is fruit waste have the potential to be used as the substrate for the production of gallic acid by solid state fermentation. These are the objectives of the study: to evaluate the potential of different tannin-rich fruit waste for the production of gallic acid by solid-state fermentation using Aspergillus niger, that's the fungus, optimization of various physico-chemical parameters for the hyperproduction of gallic acid. Material method. First of all, the microorganism was purchased Aspergillus niger from First Culture Bank of Pakistan, Institute of Agriculture Sciences, University of Punjab, Lahore. It was maintained on SDA agar and preserved at 4 degrees centigrade for further use. For preparation of spore suspension, that is inoculum, 10 ml of 0.9% sodium chloride solution was added to fully sporulated fungal culture. The spores were dislodged using a sterile inoculation loop and diluted up to 3 into 10 raised to power 7 spores per ml, and it was further used as an inoculum. These were different substrates that were used in the study. From fruit peels, banana, pomegranate, mango, and apple were used. From fruit seeds, jamun, apple, mango, and of tamarind was used. The peas and seeds of fruits were dried and ground to fine powder and sieved through the mesh of 0.1 millimeter. The powder was stored at room temperature and used as a source of tannic acid for solid state fermentation. Then the estimation of tannin was done using the method of Selval et al. Of 2011, and it is a spectrophotometric method in which the absorbance was taken at 520 nanometer after addition of ferric chloride and making the standard curve of tannin. For gallic acid production, after the estimation of tannin, 5 gram of each substrate was taken and the basil media was mixed. Substrate water ratio was set up to 1:2, pH 4 was maintained. Autoclaved for 15 minutes at 121 degrees centigrade, then inoculated 1 ml of inoculum and incubated at 30 degrees centigrade for 72 hours. The substrate giving high yield was selected for further process. After the selection of best substrate, the conditions were optimized for that particular substrate, and the conditions were substrate water ratio, effect of incubation time, inoculum volume, effect of pH, temperature, and the addition of different carbohydrates like glucose, fructose, sucrose, and starch at 0.2% concentration. Effect of addition of nitrogen source, example ammonium chloride, sodium nitrate, casein, urea, and peptone as the nitrogen source at 0.2% concentration. Finally, the extraction of gallic acid was done, and that is after fermentation, 50 ml distilled water was added and mixed slowly on shaker at 150 rpm for 10 minutes. The mixture was filtered and preserved at 4 degrees centigrade. Then gallic acid was estimated by spectrophotometric method using methanolic rhodonine at 520 nanometer. Crystallization of gallic acid was performed by using Soxhlet apparatus, and then after the filtration, the solvent was evaporated using rotary evaporator at 50 degrees centigrade. The gallic acid was estimated by FTIR, and quantitative estimation was done by HPLC using the standard of sigma Aldrich. All the experiments were performed in triplicates, and data was analyzed by using one-way ANOVA on SPSS software. These are the results of the study. The first picture shows the growth of Aspergillus niger on SDA agar plates. These are the results of the tannin content of different substrates that was used in the study. It can be seen that jamun seeds had the highest tannin content. This is the reason that from the different substrates under test, jamun seeds produce highest amount of gallic acid, that is 8.121 milligram per gram of the substrate. And then the conditions were optimized for it. At substrate water ratio 1:3, highest amount was produced. At incubation period of 72 hours, inoculum volume of 2 ml, pH 5, temperature 30 degrees centigrade. From different carbohydrate sources tested, glucose increased the yield in comparison to the control, that is 14.5 milligram per gram. Various nitrogen sources tested decreased the yield in comparison to the control. Hence, after optimization of all the conditions, 14.5 milligram per gram of gallic acid was produced. These are the crystals of gallic acid obtained after rotary evaporator. This is the result of the FTIR. Blue peak shows of the standard gallic acid, while red shows the current sample of the gallic acid extracted. From the peak pattern, it can be observed that the sample under test is of gallic acid. HPLC was done for the quantification, and 98.9% of gallic acid was quantified in comparison to standard. At the conclusion, potential of fruit waste to be used as tannin source for the production of gallic acid was explored. Production of gallic acid by Aspergillus niger was achieved using fruit waste as substrate, which is inexpensive, economical, and easily accessible as agriculture and food waste. The highest yield of nearly 14 milligram per gram of gallic acid was attained under optimal conditions, that is 1:3 substrate water, pH 5, 30 degrees centigrade, 96 hours of incubation period. These are the references of the study. Thank you for your attention.
Thank you very much, Dr. Shagufta Saeed. It was very informative talk. So our second presenter of this session is Dr. Nushin Aslam from Government College University, Faisalabad. She will present her work on bioevaluation of potentially active Joglan's regia against diabetes and atherosclerosis. Please, Dr. Nushin Aslam.
I am thankful to the organizing committee to providing me an opportunity to present my paper in the 4th International Conference on Applied Zoology 2021. My name is Nausheen Aslam. I am from Department of Biochemistry, GCUF, and my topic is bioevaluation of potentially active Juglans regia against diabetes and atherosclerosis. Juglans regia are amongst the best commonly consumed and commercially grown up tree nuts throughout the world. Consumption of these trees have been reported for many health benefits, including treatment of type 2 diabetes, reduced coronary heart disease, risk of cardiovascular disease, and the treatment and prevention of certain cancers and the reduction of different age-related nervous disorders. Pregnant regia is known as good source lipid due to their high lipid content. Their oil is used without refining for edible purposes, mostly for salad dressing. Nuts are rich source of potassium, magnesium, fiber, vitamin E, and unsaturated fatty acid, especially omega-3 and omega-6. Polyunsaturated fatty acids, which are important nutritional fatty acids. This makes Jiglin's regia necessary for a healthy diet, and as suggested by many studies, they are provide specific protection against coronary heart diseases. In history, several parts of plants, including leaf, bark, seeds, and green husk, were used in traditional medicine. Jiglin's regia is used in the treatment of diarrhea, for the treatment of tapeworm. Their oil is used. Walnuts can help in the treatment of different skin disorders such as acne, herpes, eczema, dermatitis, and itching. In folk medicine, the shell of walnut was used to heal malaria. The diminished insulin response causes defects in triglyceride metabolism, and the basal insulin causes lipid synthesis, causing lipid deposition and proliferation of smooth muscles. In patients with diabetes, hyperglycemia, and insulin resistance participate to alter endothelial hemostasis, thus favoring atherosclerotic features and coronary events. High glucose and free fatty acid level trigger accumulation of reactive oxygen species and subsequent alteration of pro-oxidant and inflammatory pathway, advanced glycation and products. These trigger inflammatory transcription program leading to overexpression of adhesion molecules, monocyte adhesion, and subsequent formation of foam cells. Metformin and simvastatin are commonly used to treat diabetes and atherosclerosis, having large number of side effects like physical weakness, uh, diarrhea, gas, headache, difficulty sleeping, um, and different. So Keeping in view the side effects, the present study was designed to check the effectiveness of selected medicinal plants, to evaluate the pharmaceutical response of potentially active plants to cure type 2 diabetes and atherosclerosis in high-fat induced atherosclerotic and diabetic rats by using plant extract. Material. The husk of selected medicinal plant was collected from Azad Kashmir, Pakistan. The bark of plant was washed thoroughly under tap water to remove dust content. The washed material was dried at room temperature until the moisture content was fully removed. Then it was extracted in 70% ethanol by such standard sock slit method. The the sock sample was filtered separately to obtain the final extract. The entire solution was filtered. The soap bar powder on the top of the top to filter paper was pressed to filter the remaining solvent present in it and then discarded. The filtered solvent was evaporated in the rotary evaporator to obtain the extract. Out of total extracted solvent, nearly remaining extract was air evaporated to obtain final bar extract. Here is the experimental design for diet and grouping of animal. Total 12-week-old albino rats were taken, having weight of 180 to 200 grams, kept in an animal house under observation in control condition for acclimatization of 1 week. The proof for all the experiment was taken from the Institutional Review Board, Government College, University, Faisalabad. Group A is normal control group, animal that were given normal diet. Group B is positive control. Group 5 rats, they were given high fat diet and normal saline as treatment like that of group A. Group C is standard control group, 5 rats. They were also given high-fat diet and standard drugs, simvastatin and metformin. Simvastatin 4 mg per kg per day and metformin 600 microgram per kilogram as a treatment. Group D is a treatment group. They also contain 5 rats, was also given high-fat diet and extract of Juglans regia 500 milligram per kg per day as a treatment. And group E, it is also a treatment group. We will say treatment group 2. They also contain 5 rats. They were also given high-fat diet and extract of Juglans regia at the rate of 250 milligram per kg per day as a treatment. At 0 day after acclimatization to blood samples from rats for their laboratory investigation, the blood glucose level was monitored weekly. Glucose was also determined, and their total lipid and their total cholesterol and triglyceride were measured. And after this, histopathology of tissue was done by the method of Chinchu Mohan and Kumar. Stained areas were observed and imaged under microscope, and now Juglans regia bark extract was prepared and its status was evaluated for functional properties against diabetes and atherosclerosis against rat model. In rat model, soil condition, ecology, climate, genotype, technical and cultural practices are the factors involved in variation of chemical and mineral content of Juglans regia. Here is the average yield, which was 14.6, and average percent yield was 7.33%. Effect of Juglans regia on body weight. The finding of research suggested that the body weight of rats started increasing from 0 day due to high fat content. The 500 milligram per kg extract of Juglans regia maintained the body weight of rats to normal level. Here is the effect of Juglans regia on blood glucose level. The blood glucose values of all groups increased due to high fat diet. As you can see in this slide, the 500 milligram per kg of Juglans regia plant extract lower blood glucose level most prominently. Then the market drug and showed minimum average drug, that is 87.6. That was the significant result. Now, the effect of Juglans regia on lipid profile. The triglyceride value of treatment group decreased significantly as compared to standard control and treatment group 1. The total cholesterol value of standard control decreases as compared to positive control and treatment control. Similarly, HDL value had nearly same value of standard control and treatment group 1 as compared to positive control. LDL value was lowest in treatment group as compared to positive group. Positive control. Concentration of LDL cholesterol and total plasma decreases by replacement of nutritional saturated fatty acid with their polyunsaturated fatty acid or monounsaturated fatty acid. Histopathological changes were seen in liver of control and castrate group animals. High power microphotographs of normal control showed fat-free hepatic chloroplasts. The liver of rats had which they were given high fat diet, showed hepatic infiltration and steatosis, high ratio of accumulated fat cells, which we say fat vacuoles, were also seen in hepatocytes. Juglans regia treatment effectively improved. Here we can see the Juglans regia treatment effectively improved hepatic fatty infiltration and inhibited liver steatosis in comparison with standard control. Juglans regia treatment significantly reduced the liver weight and liver steatosis in rats. Fat vacuoles were also not seen in these hepatocytes. The study was conducted to check the pharmaceutical value of Juglans regia extract. The result had showed that 500 milligram per kg of 70% plant extract showed highest effect on body weight, blood glucose, and lipid profiles of albino rats in comparison with standard control. Juglans regia treatment significantly reduces the liver weight and liver steatosis in rats. So we can say that Juglans regia would be a strong herbal remedy in future for diabetes and atherosclerosis. Here are the references. Thank you for your attention.
Thank you very much, Dr. Nasheen Aslam. It was very nice work. Our third speaker of this session is Faiza Zafar from Quaid-e-Azam University, Islamabad. She will present her work on evaluating the cardioprotective role of N-acetylcysteine and melatonin along with the miRNA regulation in stress-induced cardiac hypertrophy. Please, Ms. Faiza Zafar.
Assalamu alaikum, my name is Rimsha Mushtaq, and today on behalf of my other mates, Faiz and Ravi as well, I'm going to present the topic evaluating the cardioprotective role of N-acetylcysteine and melatonin along with the microRNA regulation in stress-induced cardiac hypertrophy. We define cardiac hypertrophy by defined as a thickness of the intraventricular walls, which is characterized by an increase in the size of the cardiomyocyte. But the notable thing is that though there is increase in the cell surface area of the cardiomyocyte, but there is no change in the number of the cardiomyocyte. The number of the cardiomyocyte remains the same. There are different mediators of the cardiac hypertrophy, including the cytokines, neurohormones, mechanical stress, but our main focus is to find a link as how the oxidative stress is linked to cardiac hypertrophy and how it mediates in the progression. Oxidative stress is the main regulator of the cardiac hypertrophy, and it is defined as an imbalance of the radical oxidative species and antioxidant enzymes in the body, which leads to the oxidative stress when the number of the radical oxidative species is increased. Oxidative stress is induced by the different inducers, which include the angiotensin 2, endothelin 1, as well as the isoproterenol and tumor necrosis factor alpha, etc. The pathway which is used by these inducers is different. For example, the main pathway, which is postulated to be important in progression of the cardiac hypertrophy, is known as a calcineurin and fat pathway, in which the oxidative stress plays an important role as it inactivates, or we can say deregulates the estrogen 1, which is a negative mediator of the calcineurin and fat pathway, and is regulated by the FOXO signaling. So by deactivating estrogen 1, it leads to the progression of the calcineurin and fat pathway, and when the calcineurin and fat pathway is activated under the oxidative stress, it leads to the progression of the disease, which is a cardiac hypertrophy, and which can be measured by different stress markers, for example, the BNP, KT4, etc. So the role, we also going to study and elucidate the role of the microRNAs in cardiac hypertrophy. And we define the microRNAs as a small non-coding RNA about 18 to 25 nucleotides, single-stranded, which are highly conserved and are regulators of the stress-induced cardiac hypertrophy. And they are involved in the gene expression at the post-transcriptional level and regulate it. In this topic, we are going to study the role of the four microRNAs in cardiac hypertrophy, which are the microRNA 132, microRNA 212, 152, and microRNA 142, 3P. The aims and objectives of this study was to explore the significant potential of melatonin and NAC, which are the antioxidants, and how these play a role in scavenging the free radical regulation during the pathological condition, which in our case is cardiac hypertrophy, and also to demonstrate and elucidate the importance and regulation of these four microRNAs in oxidative stress-linked cardiac hypertrophy. So material and method, coming towards study design. In this, to perform this study, we designed two models. In the first model, the ET1 or endothelin 1 was used to induce the oxidative stress, which leads to the cardiac hypertrophy, while in the other model, the isoproterenol was used as an inducer. But the notable thing is, in both the models, there was 6 same groups containing the normal group, disease group, treatment groups, and the positive control. And the dosage is mentioned in the slide. Along with these two models, we also had a third model, that was the HL1 cell lines, which were treated with a different concentration of hydrogen peroxide. So in order to elucidate that how the oxidative stress linked cardiac hypertrophy plays its role directly on heart, then how is heart affected by the oxidative stress directly. So the different diagnostics techniques were used to elucidate oxidative stress linked response, and which included the qPCR analysis and the Western blotting analysis, which were performed to check the expression level of the different markers on both the post-transcription and the translational level. Along with this, the biochemical assays was also performed for the antioxidant profiling and oxidative stress profiling. And along with this, a histological analysis was also done to elucidate the response of the disease, how the disease is progressing, and how the antioxidant treatment is controlling the disease progression. Coming towards the results. So the baseline characteristics of the animal model was, in the first one, we studied the ratio between the heart weight to body weight and heart weight to tibial length. And these two ratios, they are defined as the baseline characteristics through which we can tell whether the cardiac hypertrophy has been done or has progressed or not. So we observed that in both the model, that is an isotreated model and an ET1 model, these ratios were seen to be disturbed, as there was an increased ratio or disturbed ratio seen in the disease models, that is an ET and isotreated model, while in the groups which were receiving the treatment in form of the melatonin and AG, there was a slight reduction or a slight, we can say there was a decrease in the progression of the disease. Coming toward the histological analysis, which can be seen by the slides and also by the graph, which is showing the cell surface area, there was an increase in the cell surface area of the cardiomyocyte, showing the progression of the cardiac hypertrophy, while there was a slight decline or decrease in cell surface area in the treatment groups, showing that the antioxidants somehow play its role in stopping the progression of the cardiac hypertrophy. So to be very clear about, to get a clear picture whether the cardiac hypertrophy has been done or not, the disease has progressed or not, we check the expression of the different marker genes, and in this case we check the stress marker genes, which are the BNP and GATA4, and these are the important stress regulator of the markers which confirm the whether the cardiac hypertrophy has progressed or not. So we studied these marker gene at both the transcription and translational level, that is through qPCR analysis and through Western blotting analysis. We confirm the results, and we find out that the level of the stress marker genes in the disease group, that is ET and ISO, both in ET ISO disease group, was significantly high as compared to the normal groups, while in the treatment groups there was shown a decline or we can say there was shown slightly a decline in on receiving the treatment of the antioxidant. So we can say that the antioxidant, they played their role in somehow stopping the disease progression. Coming towards the radical, free radical analysis and antioxidant analysis, 4 different assays were performed. The Ross and the T bars were performed to get an idea about the oxidative stress profiling, and catalase and salt were performed to get a clear picture of the antioxidant profiling. And all these assays, they showed the same result, that is that oxidative stress there was an increased oxidative stress in disease groups, while in the treatment group there was shown a decline in the oxidative stress, that is there was increased antioxidant activities in the treatment groups, and a low level of the oxidative stress, showing the strong therapeutic potential of both the antioxidant, that is melatonin and NAC, in cardiac hypertrophy. So the microRNA, they play a very important role in cardiac diseases, and to analyze their expression, we've performed qPCR analysis on all these 3 models. That is the isotreated model, ET1 treated model, and HL1 cell lines, and all these 3 models showed a similar effect, that is the microRNA 152, 212 and 132. All of these 3 microRNA, they showed a significantly high expression level as compared to normal in disease groups. While contrary to this, the microRNA 142 3P, it showed a decline in the disease group, while its level was increased in the treatment groups. So in the treatment group, which were receiving the antioxidant treatment, the results were reversed. That is, the 3 microRNAs, 152, 132, and 212, their level was significantly dropped, while the level of the expression level of the microRNA 142 3P was seen to be significantly increased on treating with the melatonin and NAG. So coming towards the discussion of the paper, we can say that both the ET1 and ISO-induced cardiac hypertrophy by generating the oxidative stress. As the level of the ROS is increased, then this leads to the progression of the cardiac hypertrophy via different signaling pathways, for example, P13K, AKT, via FOXO3 pathway. However, one thing is very clear, that isoproterenol and endothelin 1 disease groups, when they were treated with the NAC and the melatonin, the known antioxidants, there was observed a significant increase in antioxidant enzyme activities, and there was a significant reduction in the disease progression. The microRNAs are known to regulate the gene expression at the post-transcription level, which was also observed by the qPCR analysis, and to which we observed that there was an increase in the expression of the 3 microRNAs, while contrary to this, there was a visible decline in the level of the microRNA 142 in the disease group. However, these results were reversed or changed when they were treated with the antioxidants, showing the therapeutic potential, as the microRNAs which were previously increase in the disease group, their level was declined or decreased when they were treated with antioxidant. And on the other hand, the microRNA 142 was seen to be decreased in the disease group. Its expression level visibly increased when it received the treatments of the antioxidant. And a very similar pattern was observed in the HL1 cell lines, which were treated with different concentration of hydrogen peroxide. So to get an idea of how the oxidative stress plays its role in cardiac cells, and how the oxidative stress is linked to the cardiac hypertrophy. So all these three models, they presented the same result, that the cardiac hypertrophy is linked to oxidative stress. In concluding remarks, we can say that the unregulated stress is the major participant in the introduction of the cardiac diseases, and the persistent oxidative stress, it regulates the anti-hypertrophic pathway elements either directly or through microRNA regulation. And the main important thing is the down-regulation of antioxidant and the upregulation of the microRNAs at the same time. They play a very important role in the progression of the hypertrophy. And we can also conclude by all the results in which the treatment group results, that the antioxidant administration, they play a very useful and important role in counterbalancing the oxidative stress. So to evaluate or elucidate a detailed understanding of the free radical roles in the cardiac hypertrophy, we can say that it has a potential to generate the therapeutic approaches for the cardiac diseases. So thank you so much. It was all on my behalf.
Thank you very much, Faiza Zafar, for sharing your work with us. Our fourth speaker of this session is Dr. Muhammad Asif Zahoor from Government College University, Faisalabad. He will present on isolation and molecular characterization of Escherichia coli harboring cholesterol resistance gene from raw milk. Please, Dr. Muhammad Asif Zahoor.
My name is Dr. Muhammad Asif Zahoor and I'm working as Associate Professor. So today's talk is Isolation and Molecular Characterization of Escherichia coli harboring cholesterol resistance gene from raw milk. Raw milk is defined by different agencies including European Food Safety Authority as milk produced by the farm animals which has not been treated 40° centigrade, nor it had any chemical or biological treatment. Raw milk is a rich source of nutrients like calcium, proteins, vitamins, and minerals, which boost the immune response and decreases the risk of many gastrointestinal disturbances. However, it could be a potential source of different pathogens, for example, E. coli or other gram-negative bacteria. The condition is reported from the developed as well as among developing countries. Raw milk is normally contaminated with different bacteria during milking process by the lacerated udder or the hands of the milker's man, urine or fecal material, or during transportation. Some of the studies describe that the raw milk is a significant or a potential source of multi-drug resistance E. coli in humans. So these multi-drug resistant E. coli or the E. coli infections are controlled by different antibiotics, and among these cholestyne is a last resort antibiotic or the last choice antibiotic among human or veterinary medicine. So no doubt the cholestyne is a narrow spectrum antibiotic, but it has developed resistance and it cannot be utilized successfully in human or veterinary medicine. A wide range of diversity is reported among the molecular levels of the cholestyne resistance, that is MCR, mobile cholestyne resistant element 1 to 9. So this is the first plasmid mediated gene discovered as MCR1 in November in China, and there are different variants and they are responsible for horizontal as well as vertical transfer from one bacteria to the second generation of the bacteria. So the frequent E. coli frequent organisms which can carry the MCR1 are E. coli, Klebsiella, Salmonella, Vibrio cholera, and the occurrence or concurrence of the MCR1 has a serious threat for the treatment of different infections. So, keeping in view the importance of the E. coli from the foodborne, particularly from the milk, so the following study is designed for isolation and identification of the E. coli and a phenotypic identification of the cholesterol resistance, and finally the molecular identification of the first variant MCR1 gene among cholesterol resistance. For this purpose, 100 milk samples were collected and processed on different agar. 100 microliter from each sample was inoculated on the agar plate, and the suspected colonies were processed. Like this is the initial and cultural characteristics of the isolates or purified isolates. So this is the typical metallic sheen color colonies on usine methylene blue agar. And here is the microscopic examination. Biochemical tests were performed as the traditional microbiology. So it's the initial data that shows the number of E. coli isolates from the raw milk. So here is the distribution of the samples, 15 sample, and we found 5, 6, 4 and 6 from different areas and the different regions. So here is the antibiotic susceptibility testing that was performed using Mueller-Hinton agar according to the standard protocol. So out of 21 tested E. coli, 4 were resistant to last choice antibiotic, that is cholesterol, and this is quite alarming. For molecular identification of the bacterial isolates, we process the E. coli isolate for DNA extraction followed by PCR analysis of the E. coli and particularly for mobile cholesterol resistant element MCR1. So single bacterial colony from each identified E. coli was inoculated into brain heart infusion broth and incubated overnight, and the growth was centrifuged at 5000 G and followed by the DNA extraction using thermoscientific genomic DNA purification kit. And the two primer sets were used. The first one is UID, that is a species and genus specific for the E. coli, and followed by the cholesterol sulfate gene amplification. So here is the amplification data of the UID gene. We confirmed 21 isolates from the 100 samples. This is a quite increase or high number. Second, we found the MCR1 gene among E. coli isolates. And here is a collective data that we found 4 resistant E. coli which are MCR1 positive, whereas 17 isolates were negative. The other data about the critical drugs, like we found 5 E. coli isolates resistance against imipenem, 1 against ciprofloxacin, and 4 against meropenem. So this is all about the current study. And finally, we conclude that proper pasteurization of the raw milk before use is recommended, or ultra high temperature treatment, and we have also educate the population about stopping the adulteration of the milk. Thank you very much.
Thank you very much, Dr. Asa Zuhur, for sharing your work in ICAZ 2021. Our next speaker of this session is Saima Firdaus from Cohort University of Science and Technology. She will present on molecular detection of antibiotic resistant bacteria isolated from goldfish, Crassius aeratus. Please, Saima Firdaus.
Bismillahirrahmanirrahim. My name is Saima Firdaus. The topic of my presentation is molecular detection of antibiotic resistant bacteria isolated from goldfish Crassius auratus under the supervision of Dr. Farmanullah Dawar, Dr. Muhammad Israr Khan, and Dr. Muhammad Qasim, Kohart University of Science and Technology. These are the following outline of my presentation, in which introduction, study gap, objective, material and method, results, conclusion, recommendation, and reference are included. Introduction. In ornamental fishes, the goldfish Charis auratus is a common freshwater fish, and they are the small member of carp subfamily. Goldfish is distributed worldwide, and the most popular culture is usually rearing in aquarium. Currently, a global increase in ornamental fish farming like Japan, Singapore, Malaysia, and Germany are the main exporter of ornamental fishes, while overall 50% of aquarium fishes are globally exported by Asian countries. The primary cause of disease in fish is bacteria, which is caused by poor management, improper transport, and poor water quality that cause direct injury in the fish. A mass mortality of goldfish has been reported in the past due to several bacterial infections. During transportation, these fishes are highly prone to bacterial infection. To avoid such infection, fishes are treated with antibiotic, which lead to develop antibiotic resistance in the normal bacteria flora. In Pakistan, especially since the last 2 decades, aquaculture is developing with a high growth rate. Almost 20 species of ornamental fishes are imported to Pakistan from the various South Asian countries. So the gap of the current study is helpful in effort of formulating the antibiotic treatment policy to control bacterial disease associated with fish. There are 2 objectives. First one is the isolation and identification of bacteria from goldfish, and the second one is the determination of antibiotic resistance in the potential bacteria. So coming to our material and method, first of all, ethical approval were obtained from Research Ethical Committee CUST. First of all, fish were randomly collected from local fish market and placed in aquarium, fishery lab, Department of Zoology, CUST. Acclimatization of fish for 2 weeks, and then dissection of fish and isolated their part like skin, gills, and intestine for the isolation of bacteria, and then further biochemical tests were performed for the identification of bacteria isolate. And then when bacteria are confirmed, then antibiotic susceptibility test of bacterial strain were performed. At the last, molecular identification of these bacteria were done. These are the collection of goldfish, and then their dissection, and then their isolated part like skin, gills, and intestine, and then serial dilution method were performed for the isolation of bacteria. Bacteria were cultured, and then gram staining process were performed. Either bacteria are gram negative or gram positive, and then bacteria were identified through different biochemical tests. At the end, pure culture were applied on specific media. Result: number of bacteria associated with different part of goldfish. These are the 6 goldfish, and we isolated their part like skin, gills, and intestine for the isolation of bacteria. Serial dilution method were performed, and number of colonies were obtained, and then through different biochemical tests, bacteria were isolated and identified. Biochemical identification of isolated bacteria from goldfish. 4 bacteria were isolated like E. coli, Pseudomonas, Salmonella species, and S. aureus, and then gram positive, in which their morphological appearance like rod, and cocci, and further biochemical tests like oxidase, catalase were performed. When bacteria were confirmed, then they are grow on a specific media like EMB, citramide agar, and MSA agar. 4 different bacteria were isolated from goldfish, like from fish 1, their organs like skin, gills, and intestine. Through different biochemical tests, 3 bacteria were obtained like Salmonella, E. coli, and S. aureus, and then the further all fishes. Microscopy of different bacteria: gram positive cocci, gram negative rod, and gram negative rod of these 4 isolates. According to the second objective, inhibition zone ratio for tested antibiotic against isolated bacteria. 4 isolated bacteria were treated with 8 antibiotics in which ampicillin, erythromycin, and vancomycin are highly resistant toward these 4 bacteria, and the mostly zone of inhibition were shown by levofloxacin antibiotic. That is the table that show the 4 bacterial isolate like Staphylococcus aureus, Salmonella species, Pseudomonas, E. coli. These 4 isolate were treated with 8 antibiotic like ampicillin, azithromycin, cefiximine, ciprofloxacin, erythromycin, kanamycin, levofloxacin, and vancomycin, in which ampicillin, erythromycin, and vancomycin are highly resistant toward these 4 isolate, and levofloxacin show high zone of inhibition toward these isolates. For the confirmation of biochemical identification of bacteria isolate, the molecular characterization were done. So first of all, DNA was extracted by standard phenol chloroform method, and then 16S RNA primer were used with sequencing. And for 16S RNA amplification, PCR was done, and primer was annealed on 56 degree. The L show the 1 kb gene rural ladder of known band were used to confirm the product size. So the first one band of ladder is 500 base pair, and the second is 1000 base pair. Third one is 1250 base pair. Fourth one is 1500 base pair, and so on. So our Amplify product was 1400 base pair. Conclusion. The excessive use of antibiotic has reduced the effectiveness treatment normally used for the control of bacterial infection and causes contamination of soil water, which lead to develop new antibiotic-resistant bacteria strain. Recommendation. The use of novel and improved drugs are recommended to prevent the drug resistance in the bacteria flora of C. aureus in order to control bacterial infection during transportation. Acknowledgement. I'm highly thankful to Dr. Farmanullah Dawar, Assistant Professor, Department of Zoology, CUST, my co-supervisor, Dr. Muhammad Israr Khan, Lecturer, Department of Botanical and Environmental Science, CUST, and my third co-supervisor, Dr. Muhammad Qasim, Associate Professor, Department of Microbiology, CUST. Thanks to all the faculty member and lab fellow. These are the references that are related to my topic. And thank you so much.
Thank you very much, Sama Firdaus, for sharing your work with us. Our next speaker of the session is Dr. Muhammad Tayyib from University of Veterinary and Animal Sciences, Lahore. He will present on locally characterized recombinant enzymatic mixture, a step for uplift of poultry feed industry of Pakistan. Please, Dr. Muhammad Tayyib.
Bismillahirrahmanirrahim. I am Dr. Muhammad Tayyib, serving as Associate Professor in the Institute of Biochemistry and Biotechnology, University of Veterinary and Animal Sciences, Lahore. So I am here with the title Locally Characterized Recombinant Enzymatic Mixture, a step for uplift of poultry feed industry of Pakistan. So basically there are two important aspects in this talk. Number one, that is the poultry feed industry, and number two, that is recombinant DNA technology. So let's talk about these one by one. So first of all, poultry feed industry. Poultry feed industry is actually an important industry of Pakistan that is committed to provide good value, good quality meat to the nation. So this industry is actually facing some sort of issues. So what sort of issues? Like the feed cost, that is the important aspect. And feed contributes approximately 77% of total cost of the poultry. So that means we have to discuss the poultry feed. And Regarding the poultry feed, basically there are two possibilities, like we can go to animal-based feed or we can go to plant-based feed. In case of animal-based feed, that is quite expensive. And while in case of plant-based feed, the raw materials are cheaper. So when we are going to utilize the plant-based feed, then we have to face the problem like plant-based feed that contain a huge quantity of cellulose, xylan, proteins, and phytate. These are the compounds; those are not being digested completely in the body of the poultry bird. That is might be due to the low production of enzyme or unavailability of enzyme, and its solution is to supplement the feed with the enzymes, because supplementation of feed with the enzymes will result in the complete digestion of these compounds, and these will provide an extra source of energy, because in the absence of enzyme, the materials like cellulose, xylan, proteins, these are not being digested and simply pass through the digestive tract with the manure from the body. And secondly, the supplementation of feed with the enzyme that is environment friendly, that will improve the efficacy of feed utilization and improve the poultry bird growth. Thirdly, this will save the huge foreign exchange, which is actually being invested on the on getting the enzymes. So this means enzyme production that is actually the solution for the complete digestion of poultry feed, and this will actually put a positive impact on the poultry feed industry. So now next question is that which sort of enzymes will be suitable for this industry? Actually, we need thermostable enzymes. Thermostable enzymes are the enzymes which will be stable at a high temperature, which can withstand a high temperature, because the pelletization step during the feed preparation that requires the treatment of feed at high temperature. Then these enzymes should be able to work in the body of the poultry bird. And so here in this study, the enzymes being produced, that is actually from Thermatoga naphthophila, that is actually a hyperthermophile. This is actually the overall strategy of working, and actually the work was started with the availability of genomic DNA of Thermatoga naphthophila. So specific genes from this genome will be amplified by the PCR so that we can have multiple copies. Then the cloning of gene of interest to cloning vector, and then shifting of that and or subcloning to expression vector. And after cloning to the expression vector, you can have the process of transformation of E. coli BL21 codon plus cells that will be utilized for the production of the specific enzymes. Then the utilization of produced protein for the purification and for further characterization, and ultimately for the utilization of these enzymes for poultry trials. So this was the overall strategy, and as a result, you can see actually the results. These are being discussed briefly. So first enzyme was phytase, which was responsible for the breakdown of phytate to its basic components, because phytate can work as an anti-nutritional factor. So the SPS page analysis, which shows the purification of this phytase, then its impact of temperature shows the optimal activity at 80 degrees Celsius. In the same way, you can see this enzyme was thermostable. Optimal pH was between 6 to 7. Then iron was actually required for the activity of this enzyme, and then the kinetic studies. Secondly, the cellulase, that is also an important enzyme which is responsible for the breakdown of cellulose to its simple basic monomeric units. So the SPS page, which shows the purification of this enzyme, This is actually a 39 kDa protein. Then optimal temperature, which was found 90 degrees Celsius, and the thermostability studies. The pH 4 is required for the optimal activity of this enzyme. Effect of metalines, detergents, and kinetic studies that was also done for the activity of this enzyme. That is, next is cellulase. The cellulase was actually purified and it was found to be a 36 kDa protein. Optimal temperature, pH, metalines impact, detergent impact, this was also examined. And then finally, the kinetic studies of this enzyme was observed. Then protease. This protease was actually of about 36 kDa protein. Here 60 degrees Celsius was actually the optimal temperature. pH 9 was actually the optimal pH. Then effect of metalines, detergents, and the kinetic studies. These experiments were performed. So after having the characterization of these enzymes was utilized for the actually supplementation of poultry feed, and feed was utilized for poultry trial. And the output of trials that concluded these enzymes actually showed a positive impact on the growth of the poultry bird. So finally, you can actually conclude that supplementation of poultry feed with locally produced enzymes, these showed a significant impact on the weight gain of the poultry birds. Thus, locally produced enzymes will be utilized as a mixture for the supplementation of industrial demands of enzymes, and this will save the huge foreign exchange for the import of these enzymes in future. Thank you. If you have some questions, you can ask.
Thank you very much, Dr. Muhammad Tayyab. It was very impressive work. Our next speaker of this session is Dr. Sadia Nawaz from University of Veterinary and Animal Sciences, Lahore. She will present on mutation in SLC34A1 gene cause nephrolithiasis. Please, Dr. Sadia Nawaz.
I'm Sadia Nawaz, and I'm working as assistant professor in University of Veterinary and Animal Sciences, Lahore. I'm going to present today my work that is related to the mutations in the SLC34A1 gene cause nephrolithiasis. Nephrolithiasis are the kidney stone formation. It is due to the solid aggregates which are formed in the urinary tract system. Nephrolithiasis affects 1 in the 11 individuals worldwide, and in Pakistan the ratio is even higher. The prevalence is even higher. We have 12 to 15% of the nephrolithiasis in Pakistan. And in our population, the kidney stones, they contain around 50% of the neurological disorders, and the kidney stone formation is a multifactorial neurological disorder which is resulted from the combined influence of the immunological, biochemical, and the genetic risk factors behind it. The genetic factors, they are being involved in the pathogenesis of the hypercalciuria and hypophosphatemia, which is related to the renal stone formation. And individuals which are having a positive family history of the nephrolithiasis, they have an enhanced risk of the experience the kidney stone in their lifetime. Kidney stones the kidneys excrete a variety of waste products which are produced by the metabolism into the urine, and the urine calcium excretion in 24 hours becomes greater than the 250 milligram per year in the females, and in males it goes 300 milligram per year, and then it leads to the hypercalciuria. Approximately 70 to 80% are the calcium oxalate and calcium phosphate containing stones which are formed in the kidneys. There are several genes which are involved. There are 30 known genes which are involved in this disease. But the SLC34A1 gene is observed previously to be showing mutations in this disease in Pakistani population. So we selected this gene, and this is a very important gene which is expressed in the apical brush border membrane of the kidney proximal tubule, where it plays an important role in the reabsorption of the phosphate. Genetic variation in this gene leads to the hypophosphatemia process, hypercalcemia, and osteoporosis. Genetic basis of the nephrolithosis have not been well defined in Pakistani population, and we focused in this study on the association of the genes genetic variability with hypercalcemia and hypophosphatemia nephrolithosis inhibition. This gene is a protein coding gene, and it is the solute carrier family 34 member 1. This encodes for the sodium phosphate cotransporter NAPI2A and plays important role in the reabsorption of the phosphate. For the study, we sampled enrolled families, and we collected blood samples, then extracted DNA after PCR amplification. We also did the sequencing, and then we performed the bioinformatic analysis. For the identification and enrollment of the families, we had taken 20 families which were having a family history of the kidney stone patients, and they were enrolled from different hospitals by taking their informed written consent, and blood was collected. Then we extracted the DNA, amplified the PCR products, and after that we did the sequencing. Our sequencing results showed that the SLC34A1 gene, it showed mutations in two families, and the sequencing results of the SLC34A1 gene showed a deletion in the coding region as C756, which was deleted, and arginine 51 was changed to GLY, and a frame shift mutation in the protein region as PARG51GLY was identified in two families, and we saw that these mutations were non-synonymous. These are the sequencing results in both of the families, and this is the pedigree of the family. We could see that both generations have affected members, and sequencing results of the other family also had shown the mutations. And if we look at the genetic outcome of this work or the overall outcome of this work, you see that this work may prove helpful to study the gene function and learn more about the pathophysiology of nephrolithosis disease. In the long-term view, this can lead to the better understanding of the genetics behind nephrolithosis that is going to contribute to improved diagnosis, risk assessment, therapeutic strategies, and to develop improved treatment models for the disorder. The results of this study will also help to provide genetic counseling to the carrier families for precaution of, and this study may lead to a more reduction in the number of severely affected children born with the inherited disorders of the nephrolithosis in the Pakistani population. And these are some of the outcomes of my work, and thank you for listening.
Thank you very much, Dr. Sadia Nawaz. It was very nice work. Our last speaker of this session is Dr. Farhan Saeed from Government College University, Faisalabad. He will present his topic on structural characterization of maize bran arbinoxylans in relation to its end-use quality. Please, Dr. Farhan Saeed.
Bismillahirrahmanirrahim. First of all, I want to thank to all the organizing committee which has been invited me to participate and to present in fourth international conference on applied zoology 2001, which is on 24th to 25th November 2021. Today I am going to present my research work on structural characterization of maize bran rapinoxaline in relation to its end-use quality. First of all, I want to introduce myself. I am Dr. Farhan Saeed, working as Associate Professor in Department of Food Sciences, Government College University, Faisalabad. And so I want to describe my research on rapinoxaline. It's the overview of whole presentation in which introduction, objective, materials, method part, result discussion, conclusion, and reference parts has been included. So first of all, you can see the maize. Maize is a cereal. Maize is alternative name is corn. We can call it corn also. So its technical name is maize. It's an annual grass. It's a overall cereals are the grains which belongs to graminea family. So overall 7 cereals are present: wheat, maize, rice, oat, barley, millet, rye. So corn is a cereal. Its leafy stalk has been inside the yellow label edible grains, third leading crops in the world, and also called as multi-purpose crop. So if we want to discuss about the kernel structure of the maize grain, it consists of 4 parts. First of all, germ, endosperm, pericarp, tip carp. 4 parts are present in any maize kernel structure. So maize bran is the byproduct of commercial maize milling process. If we want to go for milling of maize corn or maize, we call it wet milling. Okay? It contains the layers of thick cell wall. Brans contains the layer of thick cell wall, and it is originated from layers of aileron, testa, and pericarp. These are the parts of bran of any cereal. No doubt, maize bran contain higher amount of bran part instead of, you can see, millet and other cereals. So maize bran consists of our 3 parts: aileron, testa, and pericarp. So it's a low price dietary fiber. You can call fiber as a dietary fiber. It's a good source of phenolic compounds having many functional and numerous nutraceutical properties, also has many functional and rheological properties. Maize bran. So if we want to discuss about the commercial application of the maize bran, it is can be used alone or in combination with maize germ cake or meal for animal feed purposes. And maize bran is also used as an ingredient to reduce the caloric value of snack food. As you know that dietary fiber is a less caloric diet and no calories in this. So it's good in obesity, and also it has the cholesterol lowering properties. So as a filler in many food, confectionery, and baked products. So the main objective, the major objective of my research work is to extract the arabinoxylene from maize bran. Arabinoxylene are the non-starch polysaccharide. It is also called as a dietary fiber. So arabinoxylene, you can say we can extract arabinoxylene from the maize bran. It's it has a capacity to absorb 11 times more water than any other fiber. So arabinoxaline is a major part of maize bran. So second objective is to characterize the structure characteristic of arabinoxaline. I wanted to clear explanation of arabinoxaline structure and characteristics in relation to their health benefits. So third part is to observe the effect of arabinoxaline on rheological properties, technological properties, textural properties, and sensorial attributes of the product. So it's the, you can see here the research design of the presentation. You can see in first phase, I have divided overall research into 4 phases. In phase 1, extraction of arabinoxaline has been done. In second phase, structure characterization of arabinoxaline was explicated through different equipments. I will mention it in material and method parts also. In third phase, I added arabinoxaline in floor in different concentration at different levels to check the optimum level of the arabinoxaline. And in last part, I observed the effect of arabinoxaline on end-use product and end-use quality of the baked product. So it's overall research design. We can call it research design. But and in next slide, overall material and method part from maize raw material to end product. So first of all, procurement of raw material is done. 2 different maize bran varieties were procured, Pearl 11 and Faisalabad FSH 810. So both varieties procured from UB Agriculture Research Institute, Faisalabad, called as RE, and chemicals were procured from local market as well as Sigma Aldrich Company. Enzyme kit was procured from Wicklow, Ireland, and other raw material was purchased from the local market. So as well as extraction of arabinoxaline is concerned from the maize bran. So it's a flow diagram. It's a flow diagram of extraction of arabinoxaline residue fiber from the maize bran. You can see here bran part and then supernatant residue treated with different enzymes, different chemicals, and at the last we got the arabinoxaline part in pure form, in which in this process different enzymes such as, you can say, myeloglucosidase and other enzyme has been used. So for the structure characterization of maize bran arabinoxaline, Fourier transform infrared spectroscopy, abbreviated as FTIR, is used for the complete characterization of its structure. It's a treatment plan. You can see here, T node is a control treatment, no arabinoxaline added. T node is a, you can say, a whole wheat flour, and T1 is a addition of 0.5% arabinoxaline in straight grade flour, and in T2 1% arabinoxaline has been added, in T3 1.5, and in T4 2% arabinoxaline, extracted arabinoxaline has been added in straight grade flour. So it's a product the last product will was prepared with the addition of these arabinoxaline in different concentration, and then rheological properties such as phrenographic characteristics and mixographic studies have been done to check the rheology of different doughs. Phrenograph is a separate equipment, and mixograph also a equipment to use to check the rheology of flour. So for the bread, after the preparation of bread, texture analysis of bread was performed according to Pig et al. 2005 with slight modification using texture analyzer, and bread lophoism was observed through the rape seed displacement method. So these parameters were used for sensory evaluation of the product, color, flavor, texture, overall receptivity, and at the last the obtained data was subjected to appropriate statistical design for the determination the level of significance, and one factor reference was used or was performed. LSD was used for comparison of different parameters. So it's a results and discussion part. You can see here rapidons are in content in different varieties of PARL and FSH 810. In PARL, 17.52% rapidons present in maize bran. In this variety of PARL 11 maize bran, and FSH 810 bran, 15.02% rapidons was present. You can see here Fourier transform infrared spectroscopy graph, in which we see here different functional group have described here. So arabinoxyline are the major group structure group in these. So arabinoxyline consists of different phenolic compound, different phenolic acids attached with, especially ferulic acid is present and showed in this graph from FTIR. So, as well as concern the rheological properties of arabinoxyline, water absorption has been drastically increased from in T0 and in T4. You can see here in T0, 61.89% water absorption was observed, but in T4, in which 4% arabinoxyline has been added, so it's increased 61 to 69.98. It means 70%. 70% water absorption has been shown in T4, and door development time also is from 6.75 to 8.25, and door stability time 3.98 to 4.92. In control 3.98, but in T4, in which 4% arabinoxyline has been added, it's a 4.92%. And other factors, other parameter, MTI, SDI, and in mixographic characteristic, peak height and mixing time has been improved after the addition of the arabinoxyline concentration, and the best treatment was found in which 4% arabinoxyline has been added. You can see here the sensory score of arabinoxyline enriched bread. So overall acceptability of control bread, you can compare it if control to 4%, sorry, 2%. 4%, it's a 2% arabinoxyline. Sorry, I already mentioned in previous slides 4% not. It's not a 4%, it's a 2% arabinoxyline added in T4. So you can see here the sensory score in control 8.2 overall acceptability, and it decreased to 7.9 and 7.3, and in T4 it decreased to 7.3, because 2% can affect on color of the bread, because bran has brownish color, so it's decrease the color quality of the bread, but increase the water absorption, and also improve the volume of the bread. So you can see here the overall, I can conclude it is the bran is the cheapest source of dietary fiber and consists of especially antioxidant, other beneficial complexes, especially phenolic compounds. Ferulic acid is the main compound of the maize bran arabinoxyline. So arabinoxyline of maize bran contains high ratio of fiber and different phenolic compounds. The bioactive profile of maize bran arabinoxyline make it highly significant and beneficial for mankind. Arabinoxylines of maize bran tend to have positive effect on rheological properties as well as overall acceptability of the end product. So its overall conclusion and now recommendation is maize bran holds potential for helping the future generation to meet their nutritional needs. Maize bran arabinoxyline contains several beneficial compounds, functional compounds as well as nutraceutical compounds that should be added to functional food, as they tend to prevent and cure various chronic ailments, diseases, and consumers are learning more and more toward the natural remedies. As well as baking product, maize bran arabinoxyline should be added to bakery product to enhance their shelf life because antioxidant Rabinozolene contain different antioxidants, so antioxidant increase the shelf life of the baking product. Further, an excessive study is required in this regard. Novel products containing maize bran and Rabinozolene should be introduced in the market as a functional food. So it's overall, it's the reference used in my presentation. It's the overall in conclusion of my overall presentation. So thank you for your attention. If you have any question, please ask through email or through a WhatsApp group or through conference organizer. Thank you so much. Allah Hafiz.
Thank you very much, Dr. Farhan Said. It was very informative talk. With this talk, our session going to its end. Our next session will start in a while. Ms. Rabia Zara will be the moderator of the session, which will include the talks from drug discovery, biotechnology, and bioinformatics. So stay tuned, stay connected. Allah Hafiz.